Thank you for coming to this in-person environmental research seminar. I'm not sure when was the last time we had this in-person seminar, but I'm so excited that we can meet together with the special guest today. So today we have Professor Marc Weisskopf from Harvard University. He and we actually did the PhD together back to 2001 through 2005. And so when he emailed me I am visiting University of Michigan and I am willing to give a talk. That's very nice. So I'm very happy that, so I will not take up his time so he can introduce him more. But he is an environmental epidemiologist and neurobiologist. We're talking a lot about this environmental chemical toxicity on neuroscience and neurologic outcome. And then today, new study about metals in tooth and early cognitive decline. So please join me. Welcome to Dr. Weisskopf.
There's nobody.
Oh, good. I can talk for 2 hours. You guys are trapped. So, thank you, Sunjan. And am I the one that's drawing everybody like you haven't been doing in person? That's exciting. So, anyway, welcome. Thank you. And you guys are sort of the guinea pigs for me because this is a new talk of mine based on the sort of work that's been occupying my mind for the last several years, trying to get something off the ground. So it's sort of a beginning of things, and as I say, it's all new. We're trying it here, but it's also more about what we're trying to do and a little bit of data. So we'll show you a little bit of data as we go. But going to start in a way that maybe is not what you're necessarily expecting. I'm going to talk about war. So this is actually, you're probably very familiar with this recruiting poster for the U.S. for World War I and World War II. And so why are we talking about war? Well, particularly around World War II, somewhat World War I also, was really a time of sort of massive technological and scientific development. Lots of innovations and technology came, not by chance. This was a lot driven by war efforts. And part of that driving by war efforts meant that a lot of it was paid for, organized by, sort of made happen by Department of Defense and the military and things like that. So a lot of stuff came out of it. A lot of incredible inventions came out of it, but a lot of things that are potentially worrisome. A lot of pesticides started being developed around here, DDT spraying to sort of keep troops safe, right? And of course the big one around World War II that everybody thought about was the nuclear bomb, right? So these were big issues that science was contributing to and that was transforming our society and how things happen. But also partly because of the military aspect of this that was kind of driven by this, that there was also this very heavy layer of secrecy over all of this, right? The military didn't want secrets, their inventions getting out. And so a lot of this stuff was sort of done without much understanding in the general public about what was happening. And at the time, there was a lot of sort of just kind of acceptance of, science is doing good stuff. It's making all these, new technologies available to us, and we advance, and it's good for us. And there wasn't a whole lot of questioning necessarily of the role of science in this. But it was around World War II that started to change. And one of the key players in that is this guy, Barry Commoner, who in many ways was at sort of the beginning of the ecology movement and the environmental movement, which was really from its inception. In fact, he even talks about it as not really being an environmentalist, but being more of a social activist in the issues of justice and how environment and ecology were just intimately wrapped up in all of that, right? And so he wrote this book. He sort of, talked about the dangers of all this stuff that science was producing and the technologies that were coming out of the war. And he wrote, this was one of his books, Science and Survival, where he was talking about how scientists can no longer sort of just do their thing, basically, and say, well, we're doing good stuff, right? So take it. And that we had to, as scientists, go out and inform people about what was happening. And this was coming from a space of a lot of stuff was that, particularly like the effort for the bomb. Total secrecy, right? I mean, no one was supposed to know what was happening there. And one of the big issues that came out of the bomb was the concern about nuclear fallout, radioactive fallout from this. And the idea that nobody sort of knew and sort of just accepted this was really kind of anathema to him. And he was saying, we need as scientists to go out and inform the public. He was actually very involved also in the Clean Air Act of 1970 and a lot about lead, the hazards of lead poisoning. So he was sort of in on all of this stuff. And he's quoted in the book where he says that science can reveal the depth of this crisis being the one of sort of just doing science and allowing the stuff to get out there without really sort of thinking about what the consequences are. And he said, but only social action can resolve it. And he was really into the idea that, as scientists, we're experts in that science field and in the data. And what needed to happen and sort of change from what had been happening is that we as scientists had to communicate much more to the public. Let the public know what was happening and what this can do and what the stuff can do. He was then very clear, though, that at that point, there is sort of you're now moving into like moral judgment about what to do, right? The war effort, people on that side were saying, well, the Russians are doing all this stuff and if you don't develop these weapons, you're anti-American, we're going to have all these problems. And then there's the debate, well, okay, but what if there's all this nuclear fallout and people die from radiation exposure? Is that an issue? And he said, at that point, that is a moral judgment. And now we as scientists don't have any more standing than anybody else who's in the public. We become one of the public to decide what do we morally want to do. But it's our obligation as scientists to sort of say what the science is and what's going to happen. So partly in light of that and in that realm, I'm going to talk a little bit more about the work we're doing and hoping to do with a lot of metal exposures. I'm not really going into other aspects of it, but I think it's important to point out that it is a very important field, and we need to do a lot about understanding, how people get exposed to the metals. I'm going to talk about metals and health effects, and that's often what we do in epi or environmental health. You're probably all aware. There's a lot more interest and focus, as there should be now, and also understanding how do people get exposed and why are some people getting exposed more than other people. These are not random, right? There are forces acting in society that are making those things happen. So that just to say that is something that we should be doing. But back to the bomb issue, there was these really interesting debates about, and then the scientists at the Commission on Nuclear AER, I think, Atomic Energy Commission or something of the United States, and people sort of on that side were saying, listen, the fallout from radiation really isn't a problem. If you measure the radiation in the air, levels are way too low. They're not a problem for human health. Barry Commoner and a few others started an organization, he was at WashU at the time, called, I think, Nuclear Science Commission on Nuclear Information, with the key being information, and that the scientists need to communicate that. And what they were pointing out, as many of us might now be more aware of, is that it is not just a matter of the radiation fallout that's in the air, but that stuff gets on the ground and it gets into the water, and then cows eat it, and it gets into water bodies and fish, and it will bioaccumulate. And now the exposure to people can be much greater if it goes through that route. And the policy people were sort of ignoring that entirely and saying it's just what's in the air. And so it was sort of this disconnect. So they were trying to point out to the public what the issues are. So one of the very first things he did, and with others, not just him alone, this Commission on Nuclear Information, was to start a project called the St. Louis at the time it was just the Baby Tooth Survey, but they were based in St. Louis. And it has its very early origins. The idea here was you could use baby teeth to monitor uptake of metals, and in this case strontium, and in particular the radioactive isotope strontium-90, which would tell you if these kids had been exposed or were they being exposed to radiation from nuclear bomb fallout, from the testing, right, that was happening above ground in the West, in Nevada, and places like that. It's also really interesting, so this survey at the time was really interesting in many ways. The whole point of it really was to get information out and to start from engaging the public, right? So they went in fully intentionally saying, we want to work with the public, get them part of this study, get them interested in it, and have that communication between us scientists and the public. It also happened to be an incredibly, so it was, the person pictured here is Louise Reese, who was the PI, essentially. Barry Commoner was part of it too, but she was one of the leaders of this, if not the leader. And actually, it employed in terms of collecting teeth from all over St. Louis and in the end kind of the world, lots of women who were sort of otherwise hitting kind of a glass ceiling in kind of the academic world. So it was also really out front in sort of getting more women involved in science to do this. But the basic idea was if kids just gave their baby teeth, they could analyze them and see what was going on for fallout. And this is one of the cards that collect them in these manila envelopes. Kid would get a little button saying, "Gave my tooth to science," and they'd fill out a little card of who the kid was, where they were, age, where they were born, things of that sort. And the kids really got into it, right? They would send these letters, "Dear Science, my name is Kathy, and I blah blah," right? And I really want to give my tooth to science, and they want their little membership club card and their button saying, "I gave my tooth to science." So it was really something that I put this up here to show that it really did engage the public, and the kids got so excited. And because they got so excited, this started with sort of dentists and people like that around St. Louis, but kids sent in, and parents sent in teeth from every state in the United States and even other countries around the world because this became so famous. And there's one of the famous stories is one letter just got sent to the mayor of St. Louis, and the mayor of St. Louis passed the tooth on, but wrote back to the kid as the kid said, "I don't need my dime from the tooth fairy, please put my tooth to science." And the mayor mailed the kid a dime and said, "Here's your dime, I don't want you to be out anything. So use your tooth." And they did. They put this to really great use. They actually looked at the level of strontium-90 in the teeth of these kids. And so they're born in the 50s and 60s. So these are baby teeth, right? So baby teeth start forming after about the first trimester of pregnancy, and depending on which tooth, it can go up to 7, 8, 9, 10 years of age. So they could take it and look at sort of early exposure to this stuff. They had kids born, they're losing their teeth around 7, 8, 9, 10, but obviously it's giving an indication of what was going on earlier on, around the time they were born. Right around here, 1952-53 is when the first hydrogen bomb was tested in the West. And that set off a lot of above-ground nuclear testing. And so this is the early paper of Louise Reis in Science in 1961 showing the relatively dramatic increase in strontium-90 in the baby teeth of these children. This was the first paper. There weren't that many that came out of it, but it was the first of the papers that sort of were demonstrating, there's exposures happening. And these are kids largely in St. Louis, Missouri area, and this is testing in Nevada, right? So we're talking long distance transport and still getting into the food chain and getting in there. And it was absolutely critical. So this science engagement with the public and informing the public was instrumental in leading to the Nuclear Test Ban Treaty of 1963, signed by JFK, saying no more above ground testing. Now, just so you know, they didn't give up testing, they just moved it underground, but that helped with at least fallout aspects. But it sort of shows how we can really make action happen if we do environmental health right, and if we engage people and if we inform people. And this was really, the story goes that it got so much attention at some point. Louise Reis, after that publication, a little while later, was sitting at her dining room table and the phone rang and her kid went to get it, and it was JFK saying, "Is your mommy home? Can I talk to her?" and then he signed the treaty. So what about those teeth, okay? They were really successful in collecting teeth. I think they got about 65,000 in about the first year they were trying. And when all was said and done, they had about 300,000 teeth from around the world, mostly based in the Missouri area. And in a kind of fun story, I got connected to this really kind of randomly. I guess this is how science goes. But about, I'd say maybe 15 years or so ago now, literally somebody opened a closet at WashU and discovered 100,000 of these teeth in shoe boxes sitting in a closet, right? Like forgotten in the sands of time. And thankfully somebody had the idea to not just throw them away, but that maybe these could be useful in some way. So luckily enough, I got connected with a person who happened to get those teeth. And we can do a lot with teeth, right? Actually, strontium-90, of course, is a metal. That's what they collected for in the first place. This is a very famous paper from '79 of Herb Needleman. Another really interesting story, social side of science that is worth going into, but I won't at the moment for time. But he did a lot of work on the early days of lead exposure and neurodevelopment in kids. This was a paper in the New England Journal of Medicine using teeth to assess lead content and to see differences in lead content. and how that related to all sorts of neurobehavioral development in children. And pretty much across the board, whatever sort of scale of behavior you wanted to use, kids who had more lead in their teeth showed worse performance on these behavioral aspects. And you're probably well aware of it's a ton of literature now on lead exposure in childhood and neurodevelopmental issues, right? So we can use these teeth to sort of look at these exposures to metals. And of course, this is probably already on your mind, but the whole idea of the Barker hypothesis, right? Named after David Barker there from 1938 to 2013, and he sort of credited with this idea of early life exposures matter for whole life health. Using the Society now for Developmental Origins of Health and Disease, this is an example of, I think he sort of started with this idea of birth outcome, whether it's birth weight or infant mortality rates by counties in the UK, and 60, 70 years later, cardiovascular mortality. Now that's a very classic sort of ecologic type of design, so not the strongest one for epi, but that sort of illustrates the concept that he was getting at. And I have to disclaim this with some Scandinavian roots myself. I have to say that everyone forgets about Anders Forsdahl, because none of you have heard about him, but he actually basically sort of said the same thing, but I don't know, he didn't get the press that David Barker got, but he's part of it too. He's Norwegian. So lots of people are going into this idea of the early origins of health and disease, but I will say even I don't think I've actually been to one of the DOHAD Society meetings. I have been to, Philippe Grandjean runs these prenatal exposure, the PP talks. Talk to him about naming things, but whatever. His meetings are more on chemical exposures early in life, but most of those still are to a large degree focused on looking at those very early life exposures and basically childhood kinds of outcomes. For many reasons. Practically, it's just a lot easier to do. There's also really interesting work about this idea of early life exposures affecting their life health around the Dutch famine hunger during World War II, where food was cut off to the Netherlands, so there was sort of pretty much starvation conditions for women, and so depending on when that happened during pregnancy, you see really interesting effects in children later on. So this idea that the early life period is hugely important, really interesting to me. It gets even more interesting to me. Well, first of all, I should say that we know that heavy metals are important for dementias and things. I don't need to talk to this audience about this because these are your people doing this work, saying, it's out there, including some people in the audience right now. But so I will say that I think Sung-Kyung will agree here that one of the conclusions from this is there actually isn't a whole lot of work on this topic. There isn't a lot. There is some, and there's reason to suspect it, and there is some suggestion of this. But it's hard to do. But this gets me interested because I'm a brain person, so I like to know what's happening in the brain and how these things are affecting this. So the idea now, we have teeth, we can look at metals, can we look at cognitive function in some way? And it's even more, there's all these mechanisms, if you look at animal studies, that suggest possible links, not only the epi that Sung-Kyung had written about, but neuroinflammation being caused, which is relevant, oxidative stress for brain health, and even amyloid beta and things like that showing up in animal studies when you expose animals, whether it's mice or rats or rabbits, to these things. So there's all sorts of evidence suggesting metals may play a role in processes that are relevant for dementia and cognitive function, things of that sort. But as Sung-Kyung wrote, there is some epi on this, but it's not extensive. This all dovetails also with work by Nasser Zawiya's group at University of Rhode Island that he, in fact, I think Sung-Kyung may have still been at Harvard at the time when he came and talked to our group about this work that had been going on in his lab about exposing rats to lead basically only in utero to the pregnant dam here. And this is a sort of housekeeping protein in the frontal cortex of the brain, showing that you can sort of look at this housekeeping protein or gene expression, I think, in RNA in the in utero period right after birth, sort of midlife of the rat, and then in the senescent rat, you can look at the control and the lead exposed, and this is just a negative control to say, your process is working. But when he looked at apolipoprotein 695, he saw a very different picture, which was an increase in expression of that, which is an amyloid-related gene, and then one that's related to Alzheimer's, that there was increased expression around the time that the lead exposure was occurring. It actually went away, the difference between lead exposed and control in adult life, but in the sort of senescent rat started to get even stronger as a signal. And then he went on to show this for several other of these genes that are very Alzheimer's and dementia relevant, and also showed it in studies of primates and monkeys as well, suggesting that these very early exposures, even in utero, may be relevant for dementia processes in the senescent animal. And when he gave this talk, I was like, that is really cool. Too bad I could never do anything like that, because I'm not waiting 70 years to see what happens in an adult or in a human. And I was like, darn, that's so cool, but how do we do that? Well, now, having found these teeth, we might be able to do something like this. I'm putting this slide in here because it's required of anybody who talks about dementia in a talk. You have to have this slide in. You've probably seen it before. It's sort of adapted from Clifford Jack, a paper of Clifford Jack's. But basically what it's illustrating is that as we sort of look at different aspects of dementia and the cognitive aging process, it's kind of the more we look, the earlier we see that things are happening, right? So, if you want to talk about purely clinical function, when you can assess somebody, that, starts to go as you get really, really old. But if you look at sort of subtler effects on memory, and it happens a little earlier before you become kind of not able to function. If you start now doing imaging and looking at brain structure, well, those changes actually start to happen even before you notice subtle memory changes. And if you start to now look at biomarkers and things that are changing that are brain-related. This is tau-mediated neural injury, is amyloid deposits, that these things keep sort of marching earlier in time. So the idea that something early might be going on is not totally out of the question. And there's also really interesting work out of the Rush group in Chicago in the nurses' religious orders study. So one of the things that came out of that was, this was a study of nuns, right? I think around the country. I can't remember how far they went, but they basically spent their lives in the convent. Very similar lives throughout everything. But to get into the nunnery, you write essays. You have to write something to sort of as your application, I guess. And they've done study, enrolled these women in sort of longitudinal studies and follow-up, and saw who declines cognitively, who gets dementia. And then they went back to the essays that they wrote at 18 to get into the nunnery and sort of coded them for sentence complexity and idea complexity, and they can show that the higher the sentence and idea complexity in those original essays at age 18 are predictive of whether you wind up with dementia and your rate of cognitive decline in old age. So the idea that something early is happening is not crazy here. So with these teeth, we can look. So in the days of the baby tooth survey, of the strontium-90 studies, of the Herbert Needleman studies of lead, you basically just grind up the whole tooth and say what's in there. That's what the technology was at that point. Nowadays, we can actually get really fine about this, and we can cut the tooth in half. We can make thin sections of the tooth, and we can look at very specific parts of the tooth that tell us about different parts of development. So this is a incisor, a lower incisor with the top of the tooth, the crown kind of up in that direction. We've cut a thin section here. The lighter part up top is enamel, so that's the part that you're actually chewing on, and the darker part down here is the dentin underneath. This is enamel dentin junction. And if you can sort of see right around here, the black arrows are pointing to a line that's running through it that you can see in almost all teeth like this, especially baby teeth, that's known as the neonatal line. And the teeth form overall a little bit like trees, right? So you know how you can look at tree rings. If you cut a tree this way, you can count their age by the rings in the trunk. But you can basically do the same thing with teeth. I mean, it's not quite as nicely geometric as in the tree, but there are daily growth rings in teeth, and it turns out that birth, among other things, is a big stressor to the system, and that leads to sort of bunching up of these cells, and you get a line that basically marks when the child was born. So you can kind of, nowadays, instead of as I said, grinding up the whole thing, you can actually do inductively coupled plasma mass spectroscopy with a laser. So you can take a laser and kind of zap here or zap there and say, I can actually average over sort of a prenatal portion of the enamel or a postnatal portion of the enamel. What we tend to do now actually is just do a whole bunch of spots along in the dentin along this enamel-dentin junction, which kind of follows developmental time. This is early, and as you go down the tooth this way, you get to later development during the tooth. So you can kind of look at very particular parts. So we do that. We've got our early exposures. Now all we need is to do cognitive testing in older folks. We're also going to try and collect blood and toenails and water from the houses of these people. But that's skipping ahead a little bit because there's a lot of work. It turns out there was a lot of work to be done. So we have all the teeth, right? The teeth were in the closet. What we don't have is the people. So that was way more work than I ever thought. I knew it was going to be hard, but it sort of got harder at every step. Every step was worse than I had thought. So huge shout out to my RA, Kaylee McElwain, who is a whiz with computer programming and doing all this stuff. But as we started this work, the very first thing we had to do, just to show you sort of the science process, is hire somebody to sit in that garage for like a year and take all these cards with the tooth next to them and put that in an Excel spreadsheet. Yeah, exactly. So I'm glad I didn't actually have to do that, but somebody had to do that. And of course that, I thought, yeah, that's going to be really hard. But once we get that done, we're golden. We can then find the people. Well, not so much, right? So all sorts of problems would come up. You know, these cards are handwritten, decaying note cards. It's been 60 years, 50 years. So there's that issue. We don't have a unique ID for each person. And what we learned is those 100,000 teeth come from many fewer individual people. And of course, you don't lose all your teeth as a baby at the same time. So you can come in many different time periods. And on this form, right, you don't have the unique identifier. We have information like this, names, date of birth, childhood address. But of course, it can look like this, right? Somebody wrote it as Smith one time. This is the same kid, but they added an H because why not? You know, one time he's Jakey, the next time he's a little older, they call him John now, so he's John Michael. The parent's name first, we didn't have time, it's QS, that's Glenn Smith. So, and there's, getting the birth date slightly wrong, or maybe they just write 58 and not 1958. So the amount of sort of fuzzy math, logic, computing stuff that went into trying to decipher all this into first just trying to identify who are unique people in here. And we think we have basically got, out of those 100,000 teeth, about 36,000 unique individuals, with another 4,000 or so where we're a little unsure because the matching is not totally clear, and it might be one person, it might be two, and we don't really know. But we finally did that. And so we have this list of 36,000 people where we are pretty sure it is a unique person, they're not duplicated, and we've cleaned up all that information, so we have the multiple ways you could possibly write Robert, Bob, or Bobby for the names, all these kinds of things. And then we shipped all that off to LexisNexis to say, where do these people live? Where are they now? How can we contact them? Which also I thought, if we get a few hundred, this will be great, because this was in 1960, right? I mean, this was the pre-internet era. This was not electronic. So the idea that LexisNexis would somehow have been able to track these people based only on their names, maybe a parent's name, their address at birth, I was a little skeptical. I was hoping we'd get something. We've done some pilot work looking at voter rolls in Missouri and Illinois, because most of them were in Missouri and Illinois, and actually had some very reasonable success finding about a third of the people through just the voter rolls. So, we thought this would work reasonably well. And it turns out it's worked way beyond what I thought it might work. This is our recruitment so far. We started, this is July of 2021. We've got hard lines for hard mail sends and the dashed lines for email sends. Of course, sometimes we'd have an email, sometimes we'd have a physical address, sometimes we'd have a phone number, sometimes we'd have a combination of those. Clearly in this group that is I think they're boomer. I don't know. Are they baby boomers? They're probably baby boomers. Hard mail works a lot better. They don't like email so much, which is unfortunate because everything we're doing with them is electronic, so we still have to get them to the computer. But we've been now quite successful. We've got 5,000 people enrolled. These are our numbers. So we've sent out literally 85,000 emails, 75,000 hard letters, 5,000 have taken surveys, and not quite all of them are up yet to cognitive tests. I'll show you what we do with the cognitive tests. So they're now, they're still largely in the Missouri, Illinois area, but they're really across the entire country. So that was another hurdle we had to face because we can't bring them all in to do stuff. So we sort of mimicked kind of nurses' health study type approaches, and we're doing surveys with them and wound up, I'll show you the cognitive testing we're doing as well. I will say that this has been a hugely fun endeavor in many ways, in part because actually we got delayed initially because we sent out our first letters and emails, and we were so inundated with calls and literally calls, people wanting to talk for a long time about their experience. And oh yeah, I remember I gave 3. Can you look for my brother? He was in on it too. And I remember going down. So literally, that was fun until it took a lot of time. We had to find other people to help us track all those. So we had to stop the process, develop this frequently asked questions page, and say go here first. Still have a question, you can call us. So, but we would get these great calls, and I got one of the people that called. Actually, turns out she is a Emmy-winning documentarian, and she was like, "I've always wanted to do a documentary on this study. So can I?" And I was like, "Yeah, sure." So she's actually been contacting me regularly doing that. And one of the fun, let's see, in the very early, I think on the very first send we sent out, I got an email a few days after, I think, an email campaign, and my colleague down the hall said, "I just got a letter from you." Like, "Did you grow up in St. Louis?" And she said, "Yeah, I did." And I think I did too. And actually, the last letter we sent, I moved right before the pandemic, and the last letter campaign we sent out over here, I got an email from my next door neighbor from my old house saying, "Wait, I just got your letter." So these people are everywhere. So it's been a lot of fun. That part's been fun. But I wanted to get at cognitive function, and dementia's harder because obviously people who have dementia, they're probably not going to participate in the study, but we can do cognitive testing. And so I've teamed up with Laura Germain, who is a tremendous behavioral neurologist or cognitive neuroscientist, I guess, at McLean Hospital in Boston, who has developed a website called testmybrain.org. You can all go there and do these tests. And she has tens and hundreds of thousands of people around the world doing these tests online on different devices. Because one of the issues, this isn't perfect, right? In the pure cognitive world, you'd really like to bring someone in and do a 8-hour battery with them. We can't do that. If you're going to do it, I did a lot of work in the nurses' health study where one of the initial approaches people used was to do telephone interview with people. And so at least you get someone on the phone and you're doing a telephone cognitive test. I didn't quite have the budget for that here. Doing it via computer has always been sort of an idea, and even in the nurses' health study, we've been sort of talking about this, but there are all sorts of issues about what device they're on, whether they're using a mouse, is it Bluetooth, all these kinds of things. And Laura has done this incredible work, not only developing tests that sort of mimic the kinds of tests we typically do in the lab, but also with her now, hundreds of thousands of people who've done around the world, and she can know what devices they're on, what they're using it. Is it a cell phone? Is it a tablet? Is it a laptop, desktop, whatever? She can sort of account for differences and see which tests work regardless of which device you're on, things of that sort, as well as norming all this data in just a massive population. So we've worked with her, we've teamed up with her, so we're running people through a battery of tests like this digit symbol match, a graded continuous performance, multiple object testing, and sort of both auditory or visual pairs and verbal pairs, where they have to remember which, or sort of say which is, they're given a cue sky, and they're given words and say which one does that go with, or they're shown pictures and which one does that go with. So we're able to run these people through all of these cognitive tests that tap into executive function and memory and different kinds and attention processes. So that's how we're approaching the cognitive side of this. But we do have still a problem for the epidemiologists in the room, right? This is a problem with DOHAD in general. You're looking at exposures here and outcomes here. That's 70 years of time. That's a lot going on. And I have one of my, I guess I can't really call it a sidetrack anymore. It's one of my major interests is sort of methods issues in epidemiology and biases that can crop up and selection biases of who's still around to do what you want to do. And these are big issues. I came across this even more. This is my former doctoral student, now postdoc, who we did a sort of simulation piece. This was in EHP in 2021 around some weird things we were finding in the world of air pollution and autism, these strange protective effects of exposures during pregnancy, realizing that it almost certainly is a kind of selection bias related to when you have an exposure of interest that affects who gets into your study, right? So in that case, we were looking at traffic pollution or NO2, and autism was what we were trying to look at. But of course, to look at autism, you are de facto conditioning on having a live child. So if this exposure affects pregnancy loss, you can get weird effects. And if you think things are happening like this in any of your studies, by the way, we have a GitHub site with some code that you can sort of plug in these things and see, how strong do these relations need to be to wipe out an effect, cause strange effects, things like that. But the bottom line is, depending on how strong the exposure you're interested in is to the selection process and some other unknown variable that has to be related to both of these things, and whether these interact with each other at all, can drive pretty strong biases that you have to worry about. In this case, what happens when you're conditioning is you wind up setting up a negative association here. I'll talk to you later why that happens, but you wind up getting a sort of paradoxical protection from this traffic pollution. And that's what we were seeing, and we think this is what's going on. A key piece here is that the exposure you're interested in has to be related to the selection process. But this happens in older age too, right? So this is not just a birth cohort kind of thing. We could have a scenario where early metals affect whether you participate in our study at age 70, whether you're around, whether you want to participate. And if there are other things going on here, like maybe your earlier cognitive function for other reasons that affect later cognitive function and affect your participation, you can induce these kinds of biases. So it's definitely something we have to worry about and think about. We do have some big advantages in the St. Louis Baby 2 study, which is that we've got 100,000 teeth, right? So we can actually randomly take a bunch of teeth for people who didn't come back into our study and sort of use that to try and help understand what kind of, how strong this might be. We also know where they were as a child. So we can do mapping stuff and look at childhood, socioeconomic, census level types of data to also see whether those things are predicting who's coming back into our study. But we can also, in the idea of sort of looking at this, we also took a peek in a slightly different setting. Another chance thing, this is why you should go to meetings and give talks. I went to the AAAS meeting at one point and I gave a talk about this and talked about all the difficulties I was having trying to get teeth and bones as part of another study before this one that I was doing, and having a terrible time recruiting and getting adults to give me their teeth. I don't know why that was really a thing. They didn't want to. And this Donnie Stedman, who runs the forensic anthropology department at the University of Tennessee, Knoxville, raises her hand in the back of the room and says, I got teeth. And I said, don't leave until I talk to you. So she runs this thing that is actually very well known in forensic anthropology world as the Body Farm down in Knoxville, Tennessee. And they've been going, not under her direction, but under others, and she's now the lead of it, since 1981. And they now literally have like a library stacks full of skeletons. So I went down to Tennessee and we got a whole bunch of teeth, and we also brought an XRF device and measured lead in bones, in the tibia bones, sent some teeth to be analyzed in our process to kind of look at, we also linked up with NDI, so to try and find out if we could tell who had dementia or not. But even before we dealt with the dementia, we said we can just look in the controls and say, does their age at death, because this is a mortality cohort, but does their age at death relate to the lead measures we're taking? This is a tooth, and so these are all the different parts. I will say that, as I explained to you, what we do mostly with teeth is cut them in half and make a thin section. Because of other work that they do, they did not want us cutting their teeth in half, so we took a different approach here as we came with a laser without cutting the tooth at all, and we laid it down the side, down the enamel, and then down the root of the tooth. Now this is an adult tooth, so what this information is telling us, the enamel is still telling us about maybe the first few years of life. The root which is actually covered by cementum, is actually sort of applied throughout your life pretty much as the tooth forms. Not nearly as in neat a sort of timing way as the enamel, but it still sort of gives you a picture of kind of your adulthood exposure. And then we could also measure, as I said, the bones with XRF and look at lead in, that's telling us about maybe the last years to decades of life before death, of what was in the bone. So in the end, we had 110 of these donors with tooth and tibia bone lead measures. Their age of death ranged from about 46 to 97 with a mean of 75, in part because these were selected to be eligible controls for the dementia study. So that's why they were kind of skewed to older ages. And this is the kind of thing we would get. So these are the spots going down the enamel. This is for lead, so you can see, it goes up a little bit. Sometimes it goes down. Sometimes you get weird measurements. You have to decide what you're going to do with the weird measurements. But we can sort of separate them and say, let's just average what their enamel measures were, and let's average what their cementum measures were as a sort of adulthood exposure, and the enamel being early childhood exposure. And this is what we saw in these guys. So this is all 110 people, and as you can sort of see, there's this downward trend with higher lead level. This is in the cementum. So this is the sort of adulthood exposure. And in fact, if you analyze it with regression, it's about 2 years earlier age of death for a standard deviation increase in the lead in the adulthood portion of the tooth. And we really actually didn't see it with the enamel or the bone lead measure in the tibia, which is sort of much later in life, after accounting for various other exposures as well as the prior ones. So the adulthood and the late life were adjusted for the prior lead exposure. So in any case, some suggestion that there is this effect. There seems to be an association of lead exposure to early mortality, but it seems more related to the kind of adulthood measure. And for time, I won't go into all the reasons why that may be the case, but we can talk about it. So what about the teeth in St. Louis? Well, we've started to analyze them. We've actually, unfortunately, only got through about a little under 200 at this point. I think we've sent 375 so far to Dartmouth. No, we've sent almost 500, I think, to Dartmouth. We're sort of waiting to get more data. But we've actually measured about a little under 200. I'm just showing you lead and manganese here. In red is sort of our indication of limited detection of the process for measuring. So both of these are clearly well above that. We can see really good signals. Not a whole lot of variation. This is overall tooth. This is the prenatal and the postnatal period averages. Not a whole lot of variation in the lead, but pretty strong in manganese, where you see much higher levels prenatally, dropping down postnatally. And that's actually been seen almost anywhere I've seen someone present data on metals in teeth, that manganese has this very strong decline in the early years. Remember this, there are many reasons for this. I mean, this was in the 1950s, right? So lead exposures were a lot more common than they are now, and probably in these settings, they were exposed from gas and from paint and things like that. So they were pretty regularly exposed. It wasn't like there were big differences over time for an individual. Although there could certainly be big individual differences in that. So while we're waiting for, this is the part where I say it's sort of still ongoing, we're doing it, you're the guinea pigs in this. So we haven't got enough data to really dig into the tooth metal analysis. So while we're waiting, we have a lot more data on just the surveys that people are doing. And one of the things the Rush group also did, it has some data on in terms of this idea of early life being relevant, is they developed these sort of scales of cognitive stimulating activities, and in their cohorts, have asked a lot of the people these types of questions. Do you go to the library? It's a little different at age 6, right? But 12 and 18, reading books, going to library, playing games, things of that sort. And they've shown in several different studies that higher amounts of these types of activities are protective for cognitive decline in old age, dementia in old age, even the pathology of Alzheimer's in old age. So something early on is helping in some way. So we decided we put these in as well. I'll show you, you can sum these all up. They are all on like a 5-point scale, and then you can sort of average them so we get sort of a nice distribution of scores of these early cognitive activities. And I know you're thinking that I'm now going to show you about the results of how they affect cognitive activity. Well, unfortunately, the postdoc that was interested in this was far more interested in mental health than cognitive health. So I said, fine, you can look at our mental health data. So she we asked these St. Louis baby tooth participants about depression and anxiety using the Patient Health Questionnaire 9 and the Generalized Anxiety Disorder 7 questionnaires. And this was at the time we had about 2,000 some odd who had done our surveys, using sort of regular type cutoffs that people have looked at for these scales of kind of probable cases of 10 or more. We get 84 cases of probable cases of depression and 58 of anxiety, which are 4 and 2.8%, which is roughly what you see in this age population in the U.S. And so she basically asked, do these cognitive stimulating activities relate to those outcomes? This is some general characteristics of the cohort that I'm putting up here more so you can see the cohort. It is by levels of that early cognitive stimulating activity averaged over the 3 time periods. We have somewhat more, I mean, it's hard, I guess, to exactly tell. I should put the totals there, but we have a few more women than men who have done this. It is a predominantly white population, looking like sort of the areas around St. Louis at the time. This is the father's education and the mother's education. So, right, these are the parents. So we're trying to get at what is their early life social setting, childhood family structure, et cetera. I mean, here we get some distribution in kind of socioeconomics based on things like education. If we look at the participants' education themselves, it definitely skews higher. We're getting a higher education. But so when she did this, she sees actually a pretty strong relation between those early cognitive stimulating activities in childhood, averaged overall childhood, and a decreased risk of depression, not much association with anxiety. And it's this, we had some of the trouble with geocoding all our people, but in some of the people, those who could do it, you could also adjust for these sort of census level socioeconomic factors, and we see pretty much the same results. But because I also try in my methods hat to always get something on methody in here, we also looked at age-specific scores, and we did that for several reasons. I mean, one is maybe there's something about a specific age that's important, we want to know that. But there are other reasons to do it that I'll get into. But when we do that, if we just look independently at each of the scores at any of those ages, they all look relatively protective for depression, not for anxiety. But when you actually co-adjust for the other time, the scores at the other time periods, this one is not adjusted for the later ones because it's afterwards, but we see protection for age 6, not for 12, protection for age 18. Interesting kind of on its own. We also did it at age 40 where we don't see an association with either of the two. But one of the things that this illustrates that I like to point out whenever I can is the concept of negative controls in epi, right? So the idea that we don't have to just adjust and hope we've dealt with all of our residual confounding, but there are ways we can sort of explore to some degree whether any result is still affected by some kind of residual confounding, at least some types of confounding. This concept of negative control exposure is drawn in a DAG. I think hopefully everybody's sort of used to DAGs these days, but if you're interested in whether A causes Y, right, you can adjust for a set of potential confounders that you know about and have data on, right? We do that, and we hope that's it, but we're never sure whether we've got something else that we just don't know, or we know but we didn't measure, that's affecting, that's confounding our association between A and Y. So a negative control exposure would be another exposure B that does not on its own cause Y, and it's U comparable to A. So in other words, the confounding structure is the same, right? Classic example of this when this was first described is like you're interested in asthma today at the hospital and whether ozone today affects that asthma. You can put both ozone today and ozone tomorrow and ask, is that related to the emergency visits today, right? Well, the ozone tomorrow presumably is not causally related to asthma the day before, but it's certainly very correlated with ozone. So ozone on the day of the asthma visit correlated with ozone the next day. But if you do that, and this one is still showing an association with your outcome, then it's telling you you've got some uncontrolled confounding there, right? If you don't see that, it's telling you that any confounding of this type that is sort of shared between A and B is not there, right? Because otherwise you would see the association with your negative control. Whoops. Wrong way. This is what I'm basically telling you. There's a series of papers on this. These are the original ones, Marc Lipsitch. Then I wrote with some folks an extension of this because they're talking about it as, you have to know that B does not cause Y. You can also essentially take results of a study where you're not sure whether B might cause Y, but if in your analysis it's not there, then under a relatively simple set of assumptions, that still acts as a negative exposure control. And that's basically what we're sort of seeing here, is that certain types of confounding are going to presumably affect these scores at age 12 and age 18. Age 6 questions were a little different, so that might be a little different, but certainly 12 and 18. So things like genetics, right? They're not changing between age 12 and 18, or socioeconomic status that's relatively invariant. So exposures that might, or variables that might confound that are relatively time invariant, you have some evidence that they may not be acting. So I highly encourage folks to think about negative control exposures. There's also something for negative control outcomes too that you can use.
So, Mark, we are almost time. So
Almost time.
Yeah.
I will go quick. Okay, so this is just the discussion about those mental health results, so we can talk more about those. I will end by basically saying that one of the other cool things is one of the other people who contacted me after they got my letter was this chair of the physics department at University of Wisconsin at La Crosse, saying, my dean just got your letter, and I'm the chair of the physics department, and I have this podcast, and it'd be great to actually talk to you. And so she interviewed me for a podcast, as well as this is the person who's the documentarian. And so if you want to hear more about sort of the history of the St. Louis Baby Tooth survey, and then a little bit more kind of what I'm telling you now about our study, these are some fun episodes of my nuclear life. Another one that came just from people getting my letter, which was really fun to check out. And with that, I just thank my team overall, and actually this is my group at Harvard, but this is a big effort of lots of people to do this work, including Brian Jackson and Tracy Punchin, who do the tooth metal analysis at Dartmouth. Melistas Bidlak is at Forsyth, which is a dental institute in Boston area, and she's an expert on dental mineralization and growth. Joe Mangano is the guy that I got randomly connected to, who had all the teeth of the St. Louis Baby Tooth study. So they are still in his garage, now slightly more organized than they were before. Scott Bartell at Irvine for statistical stuff, and Laura Jameen at McLean. And then, of course, NIH for a lot of the funding to help support this. So, thank you. Sorry.
So yeah, we have some time. So, but if we have to leave, then
Yeah.
All right, but we have time to question.
I had a question about the early life cognitive stimulating tool that you guys use.
Yeah.
I just was curious, is there a way to know whether or not there's reverse causality going on there, where like kids that don't have lead are more likely to read for longer hours a day, and if you could control for that, or I don't know, how would that affect the data?
Yeah, so interesting question, but I think actually you've got two issues going on in there. So the issue about the lead, like that lead might affect whether you do these cognitive stimulating activities, in some cases would be sort of, in essence, if you're interested in the lead, kind of those things might be on the causal path for why you would get some problems with cognitive function or mental health later, right? That lead leads to changes in these cognitive stimulating activities that then affect your late life condition. I'd consider that a sort of mediation effect. If you're interested in the cognitive stimulating activities themselves and whether they affect that, then yeah, lead could be confounding that potentially. And so, yeah, we need to, once we get our metal measurements, we would want to look at this again and see is there an effect of the lead, and if we adjust for that, does the cognitive stimulating activities in childhood go away? That could be a potential confounding issue. Reverse causation is also something that's really a concern and something you should definitely be thinking about. And we did a couple things about that. So the same idea of the negative control exposure approach also sort of helps in this regard, in the sense that we were actually more worried about reverse causation for the score at 18, thinking that, well, okay, you're already kind of developed depression, anxiety, that's a big time period for that. And if you are depressed or anxious, maybe you do less of those things. And then that affects your later state, or you're already depressed and anxious, so you're going to report higher in old age, and it's that affected your score. So certainly something to worry about. A couple things argue a little bit against that. We didn't see this association for that same set of scores at age 40, and we sort of felt like if it was there at 18, why wouldn't it be there at 40? What's the other one? Does the not seeing it, I guess that's the one that argues more against that one, because the not seeing at age 12 may be that you haven't been depressed yet. But so the age 40 score was important there because had we seen something there, I'd be even more concerned about reverse causation issue. So it's not a perfect answer that it's not happening, but I think it's a piece of evidence that maybe it's not that. I think there was one over here. Yeah.
Super fascinating study. It's awesome. I think that it's super creative, so I'm so happy you came here and talk about it. I just have like a few questions. I'm wondering if you're considering looking at death records in the future, if that's like
Yes.
proposal number 2.
Yes, but and everyone is chomping at the bit to get me to do that, and I'm holding them off because I want to do everything we can to see if we can, how many people we can get back before we do the NDI search. We are getting some letters coming back saying this person, my spouse died, my brother died, whatever it was. So we know some of them have died. But we are going to do that. I'm just sort of waiting to exhaust our efforts to find these people.
Totally understood. Second question was actually based off of the conversations you had with people who called, like inundated you with phone calls in the beginning days. It sounds interesting to me that some of those people could indicate that their siblings participated, or that they knew their friend did also, and kind of help like in more of a community building aspect to like help you identify these other 75,000 teeth or whatever.
Right.
So I'm wondering if there's any like community organization work happening there, or to like be able to control for genetic risk based on siblings.
Yeah. So a few things, I guess. We are certainly, if anybody calls and says, my brother, my sister was this, we are like, yes, please let us know, because all this information we have from LexisNexis, one of the big issues we have is we have no idea whether it's right, whether the contact information they're giving us. So if they can give us better contact information, we'll use that. But they still have to be in our set of 100,000 teeth, right? So if they said, I did participate, but my tooth's not there anymore, we can't do anything. The community engagement there, I guess we haven't come across a way to sort of enhance the ability to find people. Another interesting letter I got actually, so I worked for the CDC for 2 years as part of a cohort, and one of my cohort classmates got one of my letters and was not a St. Louis baby tooth participant, but had just bought a house in St. Louis where she's from, and the person who had been there was the participant. And so I said, please send the letter off to that. So we do any of those, but we haven't gotten a group together that is enhancing sort of getting the word out to these folks in that way. I will say that the issue of returning results is huge, and my RA Kaylee, thank bless her heart, is on me all the time about returning results to these participants. And the cognitive results, when they do them, they get right away. Because Laura has done all this work across the world, she tells them immediately, for someone of your age and education, here's kind of where you're falling, right, in terms of everybody else. The meta levels in the teeth, we're trying to figure out the best way to return to people. And part of the concern is just that we don't really know what any individual measurement means. And so, of course, our IRB gets really worried that we're scaring people, and we're trying to say, no, I think they can handle it. We can give the proper language. But we are working with actually the Silent Spring Institute to try and use one of their sort of mechanisms, their web-based mechanisms, to try and return results so people can go in and get their own metal measurements. I said we were also collecting blood samples, and I'm working with a colleague in my department. Our hope is to look at extracellular vesicles in the peripheral blood. So this is a really exciting area if you don't know about extracellular vesicles, right? This is a neuroepidemiologist's potential dream, because getting into the brain of a live person is tough, but these extracellular vesicles get basically shot off like Star Wars pods from all cells in the body, some of them from nerve cells and brain cells, and there is the thought, and some people say they can actually determine which ones come from the brain. We're trying to, and then they bring little pieces of the cell with them, right? They're a little space pod from that cell. So we're trying to look at miRNA inside those extracellular vesicles. I'm really unsure how to return that result, because it's so like cutting edge new science. We really don't have any idea what it means. We're working on it, but we definitely are going to do that kind of stuff and try and do that. And that's why actually I've tried to be very responsive to the phone calls and making documentaries and doing podcasts, because we are letting all the people know that we're doing this stuff, so that, because the ones that call are just, they are overwhelmingly super excited about doing this again, and they remember this with great love and joy of their original participation. So that part has been fun. Yeah.
I was curious if you could talk about why there might be differences that you saw between the, what was it, the enamel which reflects, or the part of the tooth that
Yeah.
reflects adult exposure versus the bone samples,
Right.
which I believe you also said reflect adult exposure.
So this is intriguing. So the bone lead measurements, there is data out there. I don't remember whether Sun Kyung is on this paper with me or not. I don't think you're on that one. There's a bone lead mortality and cardiovascular paper, but maybe we're on others. Anyway, that there is data suggesting that lead exposures lead to an increased rate of mortality, particularly cardiovascular ones, right? A lot of that is done with blood measures, which are not great to get a cumulative exposure. We did some with bone lead, and we see pretty strong effects. But it's bone lead in cohorts that are alive. So they were measured, their measurement was made sort of in later adulthood, and then they were followed for death later. Our measurements, the bone lead of the tibia, while the same approach, it's an XRF approach, it's at death.
Right.
So it's capturing a half-life, because it's the tibia, of probably, we think, many years to decades of exposure. The problem is bone turns over now slowly, and that's why it's been a great biomarker. But as you age and as you get to later life, that starts to happen more. There's more turnover. And depending on illnesses you have and things like that, those rates of turnover and how the lead content changes can vary. So one of the reasons I think one of my concerns about not seeing it with the bone measurement is that these changes close to death are affecting what the measurement is telling us enough that it's introducing enough noise that we don't see a signal. The other possibility is that towards the very end of your life, the story's written already and the rest of the lead isn't doing much more, and the earlier lead is what mattered. And it is true that when we have more measurements on the bone than on the teeth, and when we look just at the bone mortality association in all the people we have bone lead measures on, we do see this. But when we adjust for the earlier adulthood lead, that goes away. So I'm not totally sure whether it's changes in bone turnover is making it not quite as good a measure, or whether that period is less relevant. The early life enamel part, the early childhood, is trickier in other ways. One is that's capturing a few years in early childhood, from like probably 0 to 6 or 7 or something like that. And one possibility is that period is just not as relevant as later in your adulthood for mortality. Another possibility is that's a short set of years, and the adulthood measure we have is capturing like a lot of your adulthood life. So it's just a longer cumulative marker, and this may be just the more lead you've had, the worse off you're going to be. So the longer the cumulative measure you can get is going to be the stronger indicator of it. I think that's probably pretty likely. The other possibility is that if that really early lead leads to even earlier mortality, because this sample was selected to be controls for a dementia study, so they skew older. We're conditioning on having gotten there. If the earlier life lead, in fact, was very relevant for dying early, or somehow not getting into this donor system in Tennessee, which I think is a little less likely, that could be a problem. The effect of early life lead on mortality to be strong enough to actually cause this, I'm a little skeptical of. So my biggest suspicion is that adulthood measure is just capturing so many years that it's a better indicator of really, your overall cumulative exposure, that's really, it's just the worse off you are. And it's a better measure. Okay. Great. All right.